Can everybody hear me? Is this working? Is this good? It's on? Okay, can you hear me now? All right. It always helps when you have your microphone working properly. So I'm going to go ahead and get this all started up. Oh, and the video is recording, correct? Okay, just make sure I got everything properly functioning. I know how frustrating it is if a video isn't there and you're trying to catch up on some extra notes. So let's just go ahead and get started. So I heard you just had a test, and you guys have covered a lot of information on metabolism and citric acid cycle, and that's right. Okay, so we're going to this chapter is kind of a break from that sort of lecturing that you've had. It's going to be very similar. I'm sorry, not similar. It follows up very well with metabolism, but this is a different form of metabolism. It's often referred to as macromolecular metabolism. Also, this subject is often referred to as, say, managing genetic information, or often commonly referred to as molecular biology. It's interesting to think that I might be considered a molecular biologist because at heart I feel that I am more of a chemist. But my research area actually does fall under this same topic because I deal with a little bit of DNA replication, and that's what we're going to be starting with today. As I said, DNA replication, transcription, those can all fall under category of molecular metabolism of DNA, and these involve both synthetic and degradative reactions. So we have, first of all, we'll be looking at the replication of DNA, which is basically continuing information, copying DNA and separating it so you can go to new cells and you continue with this important genetic information. Then there's the amplification of information, and that's transcription of RNA. That's turning DNA into RNA so that it can be used somehow in the cell. And applying that information is the translation of RNA into protein so that the proteins can carry out what is necessary to happen in the cell. So as I said, today we're going to start with chapter 17. This is dealing with DNA topology, DNA replication, and the packaging of DNA into the cell. And this picture is just a little picture of a virus, which shows how important it is for replication to occur, because basically the whole life cycle of a virus is dependent upon replicating the DNA, utilizing a host cell, and then packaging the DNA as well as everything else back together so that it can exit the cell. So it kind of shows a very simplified version of all these, continuing information, amplifying information and applying it, all just to help the virus replicate. So a lot of DNA metabolism, molecular biology definitely dates back to 1953 when Francis Crick and James Watson first determined the structure of DNA. I'm sure everybody's already heard this before, but we'll just review it just again, just so it's all familiar with everybody. They determined that DNA is complementary double-stranded DNA. So if we look at this structure here, you can see that there's the phosphate sugar backbone in one strand of DNA, and here's the other strand of DNA with the phosphate sugar backbones. And then you have these bases that are kind of protected inside, that are stacked between the double helices, forming what are called Watson-Crick base pairs. And you might remember that guanine properly pairs with cytosine, and adenine properly pairs with thymine. So these are what are termed the Watson-Crick base pairs. Now, when Watson and Crick first determined the structure of DNA, they recognized that there was potential for the way in which DNA replication actually occurs. What they realized is that, what they proposed was the idea that this DNA could basically be unwound and used as a template to make new DNA from the parental DNA. But it didn't solve the question about what happens with the parental DNA compared with the newly made DNA after replication occurs. So in other words, who does the parental DNA pair with after the whole process is over? There were There were two possibilities. First, there was the conservative option, where basically you have the parental DNA unzips, DNA replication occurs, and then the parental DNA repairs with itself. Or there was the semi-conservative option, where the parental DNA unzips, the new DNA is made, and actually one strand of old DNA and one strand of new DNA is paired together. And that's what semi-conservative replication would be. Well, this question was actually answered in 1958, about 5 years later, by a very classic biochemistry experiment performed by Messelson and Stahl. Basically what they did was actually a relatively easy experiment where they grew bacteria for several rounds of replication in nitrogen-15. Nitrogen-15 is an isotope of nitrogen, so it's a little bit heavier than normal nitrogen. And so since nitrogen is a very big component of the bases in DNA, it was incorporated into the DNA of these bacteria. So basically they have labeled DNA that's a little bit heavier than you might find from DNA in bacteria that aren't grown in this type of media. And what they did was, after labeling the DNA in the bacteria, they switched the media to nitrogen-14, which is normal nitrogen, slightly less dense than nitrogen-15, and allowed another round of replication to occur. And so what we have here in this picture, if you look at this A here, you can see that this is our double-stranded DNA grown. It's slightly darker blue than down here. This is supposed to represent the nitrogen-15 labeled parental DNA. SC stands for semi-conservative. If semi-conservative replication occurred, what they would see is one strand of dark blue, heavy labeled DNA, and one strand of light nitrogen-14 labeled DNA. And again, the other double helix would look very similar to that. On this side, if you see C stands for conservative. If conservative replication would occur, the nitrogen-15 labeled DNA double helix would remain paired together. So you'd have a double helix of heavy DNA and a double helix of light DNA. Well, what Messelson and Stahl actually found was, once they separated the DNA based upon density, they found that the DNA was at a density that accounted for half nitrogen-15 and half nitrogen-14. So in other words, they were observing that semi-conservative replication occurs, where we have parental DNA, that's one strand, base pairing with the newly made DNA. And thus we have semi-conservative replication where some old and some new DNA is passed on to other cells that are formed. So Messelson and Stahl is a very big experiment that is classically looked at in a lot of biochemistry classes. Now, this whole idea of DNA unzipping, base pairing with nucleotides, and making another strand of DNA, seems like a relatively easy process, right? Well, first of all, you have to take into consideration that DNA is really large. Humans have nearly 1 meter length of DNA. So it's extremely long for human genes to replicate. Also, DNA twists up on itself. Not only do you have a double helix, a lot of times these helices will twist up upon themselves, and you have a lot of different structures that you have to deal with the DNA. We'll be looking at this in a little bit when we talk about DNA topology. Also, DNA replication has to be fairly quick, because you take too long and the cell might die. And it has to be correct, because mistakes in DNA can lead to mutations, which can lead to cancer. And we'll be looking at that in chapter 18. So DNA replication isn't as easy a process as you might think. There are many enzymes and many different protein factors that interact and work together to make replication occur. We're going to look at probably a few basic ones that are commonly found at the site of DNA replication. We're also going to be looking at how DNA is packaged tightly. As I mentioned, there's a large amount of DNA that has to fit into a little tiny nucleus in the cell. So we're going to be looking some at how DNA is packaged and how it actually accomplishes making this compact DNA structure. But let's start with the basics. The first step in DNA replication is to separate the double helix. Now there's a topic in science and overlaps with math called DNA topology. Now, just like a topographical map shows you the different contours and different shapes in the land, DNA topology talks about the overall structure of DNA. If you look here, this is an example of a bacterial DNA, where you can see that this one strand right here is a double helix. You can see that it twists up with other parts of the DNA, and it forms a pretty complex structure here where there's lots of twisting and lots of coiling. This is an example of DNA supercoiling, and this is basically DNA twisting around itself. It's called supercoiling because it refers to the fact that the DNA is coiled beyond just the twisting of the double helix. Supercoiling is very useful and important for the structure of DNA because it helps to keep DNA in a form that is stable. Supercoiling results from over or underwinding DNA strands and is actually important for processes of replication, transcription, and recombination. As I mentioned, DNA topology is actually an area of study in mathematics. People can spend their whole lives studying the structure of DNA. And just to show you that this actually is an area of mathematics and study, here's an equation that is used to describe the structure of DNA, where L, you don't have to necessarily remember this, but it's just an example that L stands for what's called the linking number, T stands for how many twists are in the DNA, and W stands for the rive. So a rive is basically the number of times the helix axis crosses over itself. So here we have a DNA double helix that twists around once on itself, and this is called a rive. Now, if you might imagine that you could grab on both ends of this DNA and pull it tight, you would convert this rive into what's called a twist. And a twist measures the number of helical turns that are in the DNA structure. So you can imagine that twists and rive interconvert to tell you a little bit more about the structure of the DNA, which is important for these mathematicians who study DNA topology. DNA supercoiling, this twisting and riving, actually plays a role in compacting the DNA and helping to efficiently package it into the nucleus in the cell. And we'll be looking at that later on as well. So what does DNA topology have to do with DNA replication? Well, natural DNA is negatively supercoiled. If we were to take the DNA and unzip it, basically it's going to cause some problems, like you might see here where you have the double helix. And if you just grab these two ends and try to pull them open for replication to occur, say, you're going to end up with a very tightly compacted double helix over here, which looks somewhat unnatural for the DNA. So instead of allowing this sort of structure to occur when DNA replication occurs, natural DNA is actually negatively supercoiled. And what this means is that if you were to stretch out the DNA, it unwinds strands and makes the double helix a little bit loose, which is useful because it allows the proteins for replication access to the DNA. And this loosening of the double helix actually allows for the helix not to get all bundled up when the replication fork is formed. As I said, without this supercoiling, we would have this forced twisting here that you might see. So DNA topology maintains the supercoiling state of DNA. And this is all actually maintained by enzymes. So the role of these enzymes called topoisomerases is to keep DNA at a specific supercoiling state. And they do this by changing topology of DNA in response to other changes in the DNA. Topoisomerases accomplish this by 3 simple steps. First, they cut the DNA, and in some way they rearrange the structure, and then they seal together broken strands that they had previously cut. Now, there are 2 types of topoisomerases, obviously type 1 and type 2. And we'll be looking a little bit at each one of these. The way that I remember topoisomerases, actually often wish I had a topoisomerase every year when I get out my Christmas lights. And I have this big bundle of lights that needs to be unwound and straightened out to a long strand so I can put it on my tree, right? Well, they're always twisted up so much, I often wish that would be easier just to cut one strand and pull the big long one through, rather than having to basically unwind the whole bundle. So it'd be very useful to have a topoisomerase with Christmas lights. Also be useful for computer cords or a lot of other different applications. As I mentioned, there are two types of topoisomerases. Type 1 topoisomerases alter the twisting of DNA, and they accomplish this by forming a nick in the DNA. What this means is it cuts one strand of the DNA double helix. So if we look here, it's the double helix, and it cuts on this strand of DNA right here, forming a nick in the DNA. And this extra freedom that is allowed from this cut strand of DNA allows for a little bit of rearrangement, and then the strand is resealed back together. And you can see that the tightness of the twisting with the DNA in this picture compared to this one is a little bit looser over here. And thus, topoisomerases accomplish changing the twisting state of the DNA. Now, when the DNA strands are cut and broken, what you end up with is two strands of DNA that could be just potentially flopping around, because if you cut the strand, then you're left with two open ends. So topoisomerases often actually do form what are called phosphotyrosine intermediates. This is formed between an active site tyrosine and a strand of DNA that is cut, and it's actually a covalent link. So if you look here, this is a picture of a phosphotyrosine intermediate, where here's our phosphate, sugar, phosphate backbone of the DNA, and basically the topoisomerase would cut the backbone right here. And rather than leaving this phosphate available for reacting with anything else around, because you really don't want your DNA grabbing onto anything else around that's nearby, the topoisomerase protects this strand of DNA by using a tyrosine that's in the active site. You can see this tyrosine forms a covalent linkage to the phosphate. Basically, it helps to anchor that strand of DNA while all the rearrangement is occurring with the DNA structure. After that's finished, this tyrosine, this linkage between the tyrosine and the phosphate, would be broken, and then this strand of DNA would be reformed with the strand where it was cut from. As I said, type 1 topoisomerases cut one DNA strand, so they form one phosphotyrosine intermediate. An example of a type 1 topoisomerase in humans is topoisomerase 1. It's often referred to as topo1. And this is a monomer in humans. You can see the structure in blue here with topo1. And you can see this is DNA double helix here in the gray and orange and yellow. It's basically coming out of the plane of the picture from you. You can see that topo1 basically gives a good big hug to the DNA and encircles it. There are positive charges that are found around this area in topo1, and there's residues that would be positively charged. And these positive interactions can form electrostatic interactions with the phosphates in the backbone, because phosphate is negatively charged. So positive is attracted to negative. Thus, topoisomerase is able to facilitate binding to the DNA backbone. And in here you would find a tyrosine, which is the active site for the intermediate that's formed. Any other questions before I move on? Okay. Well, topo1 is actually inhibited by a compound called camptothecin. Camptothecin is an anti-cancer drug that binds to topo1. Basically, what this accomplishes is lengthening the lifetime of nicked DNA. So you have single-stranded, cut in a strand of DNA in the organism that this drug is applied to. And these strand breaks, there might be multiple strand breaks in the DNA, and they're very bad because replication does not occur very easily when there's strand breaks. Transcription doesn't occur. Basically, most of the functions in the cell would have to halt because these processes cannot happen. So as a result, it actually kills cells that are trying to divide. So rapidly dividing cells, such as cancer cells, would be killed when treated with camptothecin. This cancer can actually be treated with prevention or inhibition of a topoisomerase. Moving on to type 2 topoisomerases. Now the difference between type 1 and type 2 is that type 2 topoisomerases actually cut both strands of the DNA. So there's 2 cuts, thus there's 2 phosphotyrosine intermediates. You can see in this picture of a circular DNA that this double helix right here in white is cut. It can allow rearrangement, moving this section of the double helix through the break and thus reforming the double helix that was cut. It's actually kind of expensive energetically for this double helix to be moved through the cut or through the break of the DNA. And thus type 2 topoisomerases need ATP. So that's another difference between type 1 and type 2. Type 1 topoisomerases do not use ATP. Type 2 do utilize and require ATP in order to function. So as a quick review, type 2 topoisomerases cut the entire double helix, forming phosphotyrosine intermediates. They accomplish rearrangement, and then the DNA is resealed, thus altering the structure of the DNA. Now, type 2 topoisomerase that's found in bacteria is called DNA gyrase. And this is type 2, as I said. There's a drug that's actually used to treat type 2 topoisomerases, and this is called ciprofloxacin. And this drug acts on DNA gyrase. And the cool thing is that it actually doesn't act on eukaryotic type 2 topoisomerases. So it only inhibits DNA gyrase, which is in bacteria. Thus, this drug is specifically used to target bacterial infections, such as Bacillus anthracis, also known as anthrax. And this accomplishes treatment of bacillus because it either enhances the rate of DNA cleavage or reduces the rate of DNA sealing. Either way, it's not good for the cell, and replication cannot occur very easily, and the cells are likely to die. Thus, by inhibiting it, type 2 topoisomerase can actually treat anthrax. Okay. So we've looked so far at the topology of DNA, and we've looked at how this topology plays a role in basic process, or actually enzymes that are involved in maintaining this topology of DNA in order to allow basic processes like DNA replication to occur in this cell. Now I'm going to move on a little bit to talking about actually DNA replication itself. Originally, it was thought that DNA replication was accomplished through a model called the locomotive model of replication. And this is the idea that basically all the proteins involved in replication would be like a train moving along a DNA track. The DNA would stand still, and the proteins such as DNA polymerase, which synthesizes the DNA, and all the other proteins in the complex would basically move along the track of DNA, thus looking like a locomotive or a train following the track, revolving around the DNA. But it turns out that this actually is not what occurs. Once we develop methods in order to take pictures or fluoresce different types of proteins, we can actually take an image of a cell, and it turns out that replication occurs at discrete sites. You can see in this picture, here's the nucleus of a cell. You can see these specific punctate sites here that are pretty bright. Those are actually sites where DNA replication occurs. These are often referred to as replication factories. Thus, we know that DNA replication actually occurs by the current factory model of replication. what this model involves is proteins that are stationary at these foci. They stay there, they don't move. And the DNA itself is reeled through. Basically, it's pulled through real quickly for replication to occur. So instead of the locomotive model where the proteins move and the DNA stays stationary, now the proteins stay. We know that the proteins stay and the DNA is the one that's moving. These replication factories, you can see with bacteria, usually you see 1 or 2 sites where replication are occurring. In eukaryotes, you can see almost 100 to 150 different sites. Obviously, this would be a eukaryotic cell. This whole idea of factory model replication is actually very efficient for lots of DNA, which we know most cells do have. So replication factories help make replication a speedy process. Let's look at an example with an E. coli genome. E. coli have almost 4.7 million base pairs. An E. coli DNA polymerase, which facilitates DNA replication, can incorporate as fast as 1,000 bases per second. So with this many base pairs, 1,000 bases per second, that means it takes nearly 40 minutes for an E. coli genome to be replicated. Now, if there's 1 or 2 sites of replication, you can see that it is going to actually take almost 40 minutes for the DNA to be copied. Now, if you look at a human genome, there's nearly 150 million base pairs. DNA polymerase is actually a little bit slower with humans. They accomplish replication at about 50 bases per second. If one polymerase was accomplishing replication of the entire genome, it would take nearly a whole month for replication to occur. And obviously, this doesn't occur because we know that cells are replicating and duplicating themselves extremely fast every day. And in our actuality, it takes nearly an hour for the entire human genome to occur. And that's because there's these multiple factories where replication is occurring at each site. So we have nearly 100 to 150 factories where they have multiple DNA polymerases at each factory accomplishing replication. So we can easily replicate 150 million base pairs within an hour. So it's a very fast process because of these replication factories. As I mentioned, at the replication factories, there are hundreds of replication forks. Now, a replication fork refers to the point at which double-stranded DNA, the parental DNA, is separated and DNA synthesis occurs, making new DNA to pair with the old DNA. This is a picture of a replication fork. There are hundreds of these at each factory, as I said, and found at each of these replication forks would be the proteins involved with replication, such as DNA polymerase and many other factories, accessory factors that we'll be looking at today. Now, the process of DNA replication can be broken down into 3 basic steps. It's initiation, extension, and termination. The scope of today's lecture is going to look at the process of initiation and extension. So initiation of DNA replication involves the forming of this replication fork and initiating new chains of DNA, and this all begins at the origin of replication. The process of extension involves synthesizing complementary DNA. Additionally, there are leading and lagging strands that need to be synthesized. We'll look at this a little bit later, and we'll see what leading refers to and what lagging strand refers to. But basically, there's a lot going on at the site of this replication fork. There's the melting of the DNA, there's forming new DNA strands, there's a lot of different dynamic interactions that need to be accomplished. DNA polymerase cannot do all this by itself. So it needs help. It needs lots of other proteins to be there to facilitate the entire process of replication. So we're going to look at 4 proteins that are commonly found at replication forks. These are DNA helicases. And we'll look at each of these in more detail in a little bit. This is just a brief overview. You see the DNA helicases and single-strand binding protein, abbreviated SSB. Both of these proteins are involved with the initiation stage of DNA replication. They help to set up the whole situation so that DNA polymerase can actually synthesize DNA. And then you obviously would find DNA polymerase at the replication fork. And you will also find an accessory factor referred to as the sliding clamp. So to start with, as I mentioned, helicases are involved with initiating DNA replication. Their role is to separate DNA strands. It's basically, it melts or separates the parental DNA so that it's single-stranded. And it utilizes ATP in order to accomplish this. What it does is basically hold one strand of DNA and pull. So I often, and as it holds onto the single strand of DNA and pulls, it unwinds a double-stranded DNA as it goes along. So the way I often visualize this is actually with a bit of a football analogy. Now, I'm not a football expert, but I do know that there are defensive linemen that are standing on the line trying to stop the football player, the person carrying the football, from going across the line. What I picture is the DNA helicase would be the man carrying the football. And he's trying to run between two defensive linemen. Basically, what he would do is stick his head down and run as hard as he could between the two, effectively separating the two defensive linemen. So you might think of a helicase in the same way, where essentially it grabs on to the DNA and it pulls as hard as it can, essentially separating two strands, making them single-stranded. And thus it accomplishes unwinding DNA. This is pretty energetically expensive to separate DNA, so it does need ATP to accomplish its work. And it turns out that the DNA would be in this, if you look at this structure of a helicase, you can see that it's a hexamer with 6 very similar components of the protein. And the DNA would be in the center, again, coming out of the plane of this picture. And as helicase unwinds DNA, it essentially transfers the DNA from one component to another, producing a rotation that would be similar to the F1 ATPase, the ATP synthase, which I believe you looked at a while ago. And actually, it's unique, a very unique similarity between two proteins that are unrelated. Helicase is actually very fast at what it does. It can separate up to 750 nucleotides per second. And now, if you really want to, here's a pretty lame pickup line that I came across while I was studying up on this. So the next time you're at say an undergraduate meeting for people with similar majors, or just you want to try out hitting on some other person you've been eyeing in class, you could try tell them, you know, if I were an enzyme, I'd be DNA helicase, so I could unzip your genes. So. I told my husband that one, and he thought it was totally lame. He didn't even laugh. So I appreciate your chuckles. So we've looked at DNA helicases and how they can separate the DNA, helping to initiate the replication fork. Now, you might imagine that as this helicase is plowing through the DNA, there's single strands of DNA that are left behind that could be flopping around, available to interact with many different things. What could potentially happen is that these single strands would actually reanneal with each other and reform the DNA. Or they might interact with themselves and form some sort of secondary structure with the DNA, or with single strands. Either way, whether reanneals or secondary structure occurs, DNA replication actually isn't very easily accomplished if this structure is present. It actually inhibits replication because it's abnormal structure for DNA polymerase to handle. So we have single-strand binding protein, SSB. Now, a wild guess, what does SSB accomplish? Well, obviously, it binds to single-strand DNA. And basically, it acts as a coat for single-stranded DNA to prevent this formation of secondary structure with single strands or re-annealing of the DNA. So as DNA helicase is unwinding, you might imagine that these SSBs are waiting around to just hop onto the DNA to keep anything else from occurring other than what you want to occur, which would be DNA replication or transcription. And there's an important lysine on SSB, which can be positively charged at physiological pH, and that is there to interact with phosphates in the DNA backbone. It's not very specific, so basically it can interact with any phosphate, thus coat the DNA just about any time there's single-stranded DNA found. And you will find that single-stranded binding proteins are involved with almost any form of DNA metabolism. Replication, transcription, recombination, just about anything you might think about involving DNA. And finally, we're getting to DNA polymerases, which do the actual act of synthesizing DNA. They accomplish this by incorporating nucleotides, utilizing the old or the parental DNA as the template. And using this template, they can base pair with incoming nucleotides. Now, here we have an example of a linearized example of DNA. Here you can see this 3' strand of DNA. The line represents the DNA strand. This would be the parental DNA, which is also called the template. Now, for reasons that we will go into in a little bit, DNA polymerase requires a primer to be present on the DNA. So this strand, the shorter strand up here, would be what's called the primer DNA. DNA polymerase utilizes this primer and incorporates the correct nucleotide utilizing base pairing in order to synthesize new DNA and make this primer strand longer. So you can see that the next base in the template is a T. So that codes for incorporation of an A because thymine base pairs with adenine. So it incorporates DATP to make this primer strand longer. Here you can see the base pair that is formed. And so this primer strand is growing. Now, DNA polymerase actually accomplishes this at a very fast pace. And it's because of some other proteins that are involved, which we'll see probably in the next 2 slides. So you might notice that I'm going to flip back to the last slide. You might see that I notated 5', 3' ends. Now, just as a review for the structure of DNA, there are these numbers, the 5' and 3' refer to the carbons that are on the sugar backbone. So if you remember, nucleotides are composed of a phosphate, sugar, and a base. These numbers refer to the carbon number on the sugar. So here we have deoxyribose. Here's carbon number 1, 2, here's carbon number 3. So the 3' OH is sticking off of the 3rd carbon. And the 5' end of this structure is at carbon number 5. So here's 4 and 5. You can see that the 5' end of the nucleotide is attached to a phosphate, which is attached to the rest of the DNA chain. If this were the last nucleotide on the chain, this would be the 5' end with a phosphate. So DNA polymerase synthesizes DNA in the 5' to 3' direction. And what this means is that it uses this 3' OH to add on the next incoming DNTP, or nucleotide. What happens is this 3' oxygen of the OH group basically attacks the phosphate on the incoming nucleotide, the one that's going to be incorporated, forming a covalent bond between the 3' carbon and the 5' phosphate of the next base. And then you lose these 2 phosphates in the form of pyrophosphate. Thus, you can see that it helps to extend the length of the DNA and leaves another 3' OH available for incorporation of the next nucleotide. Are there any questions about this? Yes? Oh, really? figure out the numbers, it takes 80. Well, if you assume that maybe there's 2 replication sites, 2 factories that are occurring, then you can cut it down to 40 minutes. I got to check my math next time. Okay. Alice?
I'll just mention that the bacteria can replicate anywhere from 20 minutes for a whole round to a month. And so it's a great deal on organisms of circumstances.
All right. Okay, so back to DNA polymerase. We're going to refer to, DNA polymerase is often referred to with this term called processive. Basically what this means is that the polymerase is able to bind to the DNA and incorporate many nucleotides at one time before it ever leaves or dissociates from the DNA. It turns out that the processivity of DNA polymerase is actually better in vivo than in vitro. So what I mean by in vitro is if you take a test tube and you just throw in DNA and your DNA polymerase, you see that it actually is fairly processive. It might incorporate anywhere from, say, 5 to 50 nucleotides. But if you could measure processivity in vivo, it would be a lot better, and it could incorporate thousands of nucleotides before ever leaving the DNA. And that's because of the last protein factor that we're going to be looking at in this lecture, and that's called, and those are referred to as sliding clamps. And these sliding clamps help to enhance the processivity of DNA polymerase. Well, what is sliding clamp? It's referred to as a sliding clamp because basically the protein clamps around DNA. You can see this is a simple schematic of what a sliding clamp looks like. You can see in blue that these 3 different subunits help to basically encircle the DNA. And it also interacts with DNA polymerase. So you might imagine it holding onto the DNA and grabbing onto the DNA polymerase and saying, okay, do your job. So the polymerase is basically associated with the DNA, and thus it enhances how long the polymerase is associated with the DNA, thereby increasing how many nucleotides it can incorporate at one binding event. You can see here are 2 different structures of DNA polymerases. This one has only 2 subunits, and this one has 3 subunits. But regardless of the structure, they both have similar function, and that's to basically clamp onto the DNA and hold the polymerase on, so replication can occur in a very fast and processive reaction. Now, I think, well, let's just look a little bit at this. Now, we're returning to DNA polymerase. It turns out the structures of DNA polymerases are all actually very similar, although the sequence of the amino acids in the proteins are very different. All the structures resemble what you might think of as the right hand. Basically, it's there ready to grab onto DNA and accomplish replication. So here's the structure of a DNA polymerase from E. coli. You can see here in red would be the fingers of the right hand. The green represents the palm. And then this yellow or orangish color would be the thumb of the DNA polymerase. So you can see that easily across this palm you might be able to lay the DNA, and thus it's able to grab onto the DNA and incorporate nucleotides. So the palm does hold the DNA and also holds magnesium, which you'll find that magnesium is actually very important for a lot of proteins that bind to DNA. And magnesium helps to facilitate replication and is necessary for the process. The fingers and thumb, their role is to grab the incoming nucleotides and hold them in just the right orientation so it can be incorporated to the primer strand of the DNA. So you might picture that it's just this hand that would be grabbing nucleotides, incorporating, opening and closing over and over, and that's essentially what we picture happens from kinetic studies and some other structural studies of DNA polymerases. Basically, it's just this opening and closing and incorporating and moving along the DNA thousands of times per second, even if you look at DNA polymerases in E. coli. I think I'm actually going to stop there. It looks like we might be a little bit early, but it's a good stopping point for today. Okay, I'll do all that. So we'll just